Hi, my name is Joseph Sullivan. I'm a professor here at Marietta College, and I want to welcome all of you to College 194, Pioneering Through a Pandemic. We're very excited that you have decided to join us in this class. We'll be meeting from now every Monday through July 24th, and every week you'll hear from a different expert in a different area, all about different disciplinary responses to pandemics such as the one that we're currently experiencing. My purpose in here to greet you is, first of all, welcome you to the class. And then secondly, I'm going to talk to you a little bit about the structure of the course and why we have arranged this particular type of experience as a lead-in to Marietta College for you. And so I'm going to share my screen. And what I would like to start with is a quote from the article that I've posted on Moodle. And this is from Lynn Pasquarella, who is the president of the Association of American Colleges and Universities, which is a consortium of all kinds of schools, community colleges, liberal arts schools, research universities that really come together to decide what does college mean if so that if you go to any college that there is some type of tangible experience that employers will know that you received, right? And so every 5 years or so, the association, the AACNU, does a major survey of employers to find out what is it that people who hire graduates want from new employees. And this quote is from an article from the Harvard Business Review that is linked to Moodle for you. And what they value the most is in their students' undergraduate experience is how well the experience advances critical learning outcomes. 80% of the employers want to see that primarily. So we have designed your Gen Ed to follow these different components. And so what you see here is a list of different parts of your college education. I've highlighted a couple of them. Number one, knowledge of human cultures in the physical and natural world. That means all the different content areas that you study. And over the next 7 or so weeks, you're going to be hearing from somebody who does microbiology tonight. You'll be hearing from people who do psychology, people who do communication, graphic design, history. All of those different disciplines are things that people should study during their college experience. And then at the very bottom, you'll notice that I've also highlighted integrative and applied experience, which means that that you get rid of this, so that you're able to integrate and combine those different areas of study. And to that end, there is a reflective essay that you'll be writing at the end of the course that will allow you to synthesize the different things that you learned during the next couple of months of studying. I wanted to show you the Gen Ed audit sheet. This is a list, basically a document that we use. I'll scroll up so you can see it. For all students here, you kind of check off as you take various required classes, and you notice that we have a major that you need to do, a distribution of classes. And when you look at the list of different types of classes here, I've highlighted all of the different disciplines that the speakers are coming from over the next few weeks. And so you'll see that every one of those categories has a couple of speakers in it. And so you'll get a nice general introduction to the different areas of study here at Marietta. And as was mentioned before, there is a reflective essay that you'll be doing at the end. And I wanted to show you the prompt. This prompt is on Moodle already. But I just wanted to introduce it to you. So you'll notice that it's split into two different options. You're only going to do one. And so you can read them both and decide which is better for you. I'm going to primarily read the top one and then tell you how the bottom one is a little bit different from that one. So in option one, you are going to synthesize, which means to draw connections between a number of the different evenings of speakers that you will listen to and engage with over the next 7 weeks. You're going to write a 3 or 4 page paper that reflects on how those different disciplines help us as an educated citizen understand pandemics, right? And so you'll take that essay and upload it to Moodle by the end of the course, which is Friday, July 24th. The second approach would be to, instead of just writing an essay, to do some sort of creative project. We have a graphic designer who will be one of the speakers, so you could do a graphic illustration, a painting, poem, or something like that. There still would be a reflection essay that goes along with it, primarily to explain what the choices you made were in that creative project. If you do that option, it would also be due the same day. I mention this now because over the next few weeks, you might want to begin, as you're listening to these different speakers, to think about what you might want to write about. You will not have to write about all 7 of the different speakers. Instead of, there's actually 8 speakers, but the last week is a returning of a couple of the earlier speakers. Instead, you have to mention at least or discuss at least 3 of them. So about half of the course, half of the evenings you'll have to address in the reflection essay. We're in week 1 now. I thought I would just tell you about this prompt. I'm going to come back at the beginning of week 4, and we'll spend about 10 or 15 minutes at the beginning of that evening sharing some early thoughts on approaches you might make to that project. And then I'll be back the last week of the course to talk through some of the logistics of how you might get that wrapped up, written, and turned in. So I'll be answering questions in both cases, either as a brainstorming function or to kind of help you figure out how to turn in the paper or project if it's some other kind of thing. In any event, so tonight I'm going to introduce to you Steve Spilatro. Dr. Spilatro is in the biology department here at Marietta College, and he's going to be beginning this series by talking about virology, or the study of viruses. And obviously that is at the heart of this current crisis that we're suffering as a world. And Dr. Spilatro will also be back in week 8 to talk with us. So welcome to the class. Looking forward to listening to Professor Spilatro tonight and learning along with you all about the biology of this type of thing. So I'll turn it over to Steve and stop my share.
Okay. Okay, here I am. Well, thank you very much for that nice introduction, Joe. And I'm really pleased to be here today to talk to everybody about our current pandemic. And this is certainly interesting times that we're living in from so many perspectives. And it gives us the opportunity to talk about connections between many different disciplines. So what I'm going to do is I'm mainly going to be presenting a PowerPoint presentation, and I'll be welcoming questions that people may have as we go along. I have with me my requisite cup of coffee. A good cup of coffee gives me pause, literally, to take a break and catch my breath and to give the opportunity for people to ask questions. So I may be sipping from that periodically as we go along. And so I'm going to share my screen here in a minute to um, get my PowerPoint presentation underway. And that should be this one here. Okay, so yes, we're going to be talking about virology with a focus on COVID-19, the pandemic named for the coronavirus that is the agent of this disease that was first identified in 2019, and thus the name COVID-19 for the disease. Certainly, we're all aware of the enormous rate of infection and the number of deaths that have resulted from this pandemic. And we're going to be talking about a number of different subjects that relate to that. And so, and just to kind of give you an idea of what some of the topics are that we'll be discussing, it includes what are the properties of the virus that causes COVID-19, and talking about the properties of viruses in general and how the SARS coronavirus type 2 compares to some other viruses. How does the virus spread? How does it infect cells? How does it cause the symptoms? How does our immune system respond to the infection? And that's important to understand. We're going to put that in context of the potential for developing a vaccine that will hopefully allow us to control the spread. And so in that context, also what will be some of our options for controlling and treating the disease. We'll see how far we get. As I said, I do welcome questions, and I will take time to answer those. And if we don't get through everything, then that's fine. I will be back, as Joe mentioned, for week 8 to do an update, see where things are after about 2 months' time. So, well, let's sort of put it into a broader context. Why a pandemic, and why now? And for many people, it's maybe not that big of a surprise. Epidemics and pandemics have been common for most of the history of human civilization. Hell's Triumph of Death, which he painted in 1562, conveyed a world seemingly devastated by death. And so it must have seemed when bubonic plague, smallpox, cholera, other diseases would strike with regularity, probably about every 20 years. Once in a lifetime, pandemics would ravage through the cities and villages, oftentimes killing 20% or more in the great bubonic plagues. As many as 50% of human populations were killed. In 1582, they did not know, nor would they have believed, that their misery was caused by bacteria and viruses invisible to the unaided eye. The microbial theory of disease dates back only to the mid-1800s, which left people at earlier times to attribute such things to divine retribution for some perceived sin, probably somebody else's, of course. But when we look back at history, when we study literature or even art, and certainly science, the occurrence of the COVID-19 pandemic is not unexpected. It was just a matter of time. Maybe the big surprise has been that it was the coronavirus, since influenza has been a common epidemic a number of times in the last century. Well, we now know that numerous bacteria and viruses are pathogens. Not the majority. Most bacteria are innocuous. We have a tremendous microbiota that are part of our normal functions that are essential to our growth and development and our normal physiology. But there are a fair number of bacteria and, of course, viruses that are pathogens. Scientists fear most viral pandemics, such as influenza, and of course now COVID-19. Why? Why do viruses pose such a threat? Well, let's begin by talking about what is a virus.
Steve, we have one question, if you would.
Okay. Okay.
We had somebody who wanted to know what does it mean that it's a pathogen?
Great question. And actually, we have a number of terms for the organisms that cause diseases. One of them is to call them a pathogen. So a pathogen is something that causes a pathology, a harm to the body. Sometimes we refer to them more generically as an agent, the agent of a disease. When we want to be more specific, we might refer to the pathogen or the agent by the specific type of organism that it is. That it is a bacterial pathogen, or that the agent is the influenza virus. So a pathogen refers to the type of entity that causes a particular disease. Okay, thank you. Okay, so what is a virus? It's remarkable, to be quite honest. It's a tiny biological device for replicating a genetic code. That's what it does. That's the only thing that it is designed to do, to make a copy of its genetic code. But it can only do that inside a host cell. It cannot replicate outside independently. It must have a host cell in order to replicate. Inside, the virus turns the host cell into a virus-making machine, taking over all or many of the cellular functions and putting them toward the replication of the virus. The host cell is destroyed as a consequence, and this and other consequences cause illness and possibly death. Where do viruses occur in the tree of life, one might ask? And here's a big tree of life. Let's look, shall we? Let's scan up. Well, there's people, we got all sorts of animals, fish, plants. Down near the bottom here, we've got invertebrates. Way down at near the bottom, I can see some bacteria. But I can look as deep and far into this tree of life as I want, and I will not find viruses. Why? Because viruses are not alive. Now, bacteria are that other important category of pathogens that I mentioned before. Bacteria cause a lot of common illnesses, responsible for certain types of pandemics. The bubonic plague was caused by bacteria. Cholera is caused by bacteria. But bacteria, even though they're very small, are cells. They have basic cellular structure. Cells have a cell membrane. They have DNA to hold their genetic code. There's cytoplasm inside the cell. That's metabolically active. It's doing things. There's enzymatic processes occurring, and energy utilization is occurring on a continuing basis. And there's storage structures even in bacteria. And bacteria have certainly one of the simplest of cell structures. Viruses are not cells. They don't do any of those things. They lack cytoplasm. They lack metabolism. A virus, when it's outside of a cell, is basically an inanimate object. I sometimes think of it like a bullet. And a bullet sitting on a desktop, on a counter, does nothing. It is an inanimate object. But when it's activated, it certainly has some destructive power. Likewise, a virus outside of a cell, outside of the specific type of cell or cells it can infect, is doing nothing. It's only able to replicate its genetic code inside a host. Okay, how small is a virus? Well, we've got a number of ways of comparing it. And one of the most difficult things for people, us, humans, to appreciate is how small viruses are. Their size is so far outside of the normal range of sizes of things that we're used to thinking about that we have difficulty really perceiving it. And we've got a number of ways of trying to convey it, but still, I don't know if even I can truly say I absolutely understand how small a virus is. But we can try. Now, what I've got here is a picture that shows several types of cells and some viruses. And so my cursor is running along the cell membrane of a liver cell, a typical human body cell. And if we were to show this entire cell, well, it would be extending up off of my computer screen, up along the walls of the room I'm in, and probably up to the second floor, okay? Well, we can't show the whole thing, but we can see part of a liver cell. Now, superimposed over that part is a typical bacterial cell, Escherichia coli, very common organism, part of our digestive tract microbiota. And a typical bacterial cell is about 1,000-fold smaller than a typical body cell. Now, superimposed over the E. coli is a variety of different types of viruses. And they range in size, the largest being about 200 nanometers or so in diameter, all the way down to some of the smallest, such as a poliovirus, which are about 1,000-fold smaller, again, than a bacterial cell. So one way to think about the relative sizes is that a bacterial cell is about 1,000-fold smaller than a typical body cell. A virus, the smallest viruses are about 1,000-fold smaller than the bacterial cell. And it's so small that it can only hold genetic information and maybe a few enzymes. It can only hold what is necessary to get it into a host cell so that it can replicate using the host cell machinery. Well, I've got another little tool to allow us to see the relative size of a virus. It's a little animation, and so I'm going to switch my screen share, at least I'm going to attempt to here. and show this animation. So bear with me, and let's pull up this animation. Okay. Okay. So this animation is going to allow us to step down through the size of some things, organisms that you may be familiar with, to compare their relative sizes. Okay, got some sound effects to guide us along the way here. Now, what we've got is the head of a pin that has on it, and you probably can't see it at this point, a hair, a human hair. I don't think there's any angels on it, but there is a human hair going across there. And what I'm going to do is increase the magnification, and we're going to see the relative magnification along this scale here as we go along. And what we're going to see as we get smaller and smaller is some small animals, some pollen, some human cells, yeast cells, bacteria, and then down to some viruses. So let's zoom in here and let's see what they look like. Okay. Everything's better with beeping sounds. Okay. So there's a hair sitting on the head of this pin. Oh, we can start to see something else on there. A little dust mite is sitting on it. But we're at about a 10-fold magnification. Okay. Dust mite. Oh, it looks like there's another one hiding behind the hair over there. And we can just begin now to see something else, probably a little pollen grain. Okay, 1,000-fold magnification. We can see the pollen grain. We can see a lymphocyte, a white blood cell, red blood cell. That's a yeast cell there. Now, 1,000-fold magnification is the highest magnification possible with a common light microscope. So this is the type of microscope you would use in a common laboratory with the light source down below. You might be looking through a microscope slide at something. But the maximum, the theoretical maximum magnification is about 1,000-fold. So to see more detail than what we see here, it takes some much more sophisticated types of microscopy and some electron microscopy. And we'll be looking at some of that later on as well. But let's continue increasing our magnification. Okay, so remember, pollen grain, we've got a white blood cell, red blood cell, yeast cell, and now we can start to see some bacteria. Okay, we got probably some staphylococcus, maybe some E. coli, typical prokaryotic type cells with that very simple cell structure. Okay, 10,000-fold magnification, 2 micrometers is about the dimensions we're looking at here. Okay, now there is one of the largest viruses. Whenever a virologist sees that shape appear in a cellular sample, it sends shivers up the spine. That little loop at the end is a dead giveaway for Ebola. So that's an Ebola virus right there, but it's one of the largest ones. But if we want to see the rhinovirus, we have to go upwards of several 100,000-fold magnification. That's how small it is. So let's kind of try to put that back in perspective. As I said, the smallest type of viruses are about 1,000-fold smaller than a typical bacterial cell. Typical bacterial cell can be 1,000 times smaller than a typical human cell. And these are actually kind of small on the size of body cells. The blood cells tend to be on the small side. Which all is in a whole nother universe of size, so to speak, relative to multicellular animals such as a dust mite. Okay, well, maybe that helps give us an idea about the relative size of viruses and all the more wonder, if not all, at what they are capable of doing. So let's return now back to the PowerPoint. Oh, there we are. Okay.
Dr. Spilatro, while you're doing that,
Yes.
has asked the question, can bacteria cells host viruses?
Great question. Great on a number of accounts, and actually they can. The other name for a virus, sort of the technical jargon type name, is a phage. So if you want to sound really sophisticated, you refer to them as a phage. And so viruses that infect bacteria are typically referred to as bacteriophages. Phage meaning to eat, bacteria eaters. And it's also interesting because, in fact, some of the first viruses that were discovered were viruses of bacteria, as some of the great microbiologists of the golden age of microbiology, Pasteur and Koch and others, suddenly, periodically, their bacterial cultures would suddenly die on them. And they finally discovered that there was some invisible agent that was causing the bacteria to all die. And it turned out that it was bacteriophages. So yes, indeed, there are viruses that infect bacteria. Any other questions? Okay, let's go back, but let's get our PowerPoint up and running again. Okay. Okay. Okay, there we go. Lots of clicking to get from one place to another through our screen sharing here. Okay, well, let's press ahead now and talk about the typical structure of a virus. And as I said, they can be very simple, and most of them fall into just a few common basic structures. And the simplest of the viruses consist only of a protein coat called a capsid, made up of some number of individual proteins that come together and form an enclosure for the genetic material. A common shape is an icosahedron, a 20-faced structure made out of triangles, a very simple but very strong structure that humans rediscovered. Buckminster Fuller put forth the design of the geodesic dome that we still see used periodically today because of its simplicity of structure and its great rigidity. Well, nature discovered this design quite a while earlier, and the other common structure is a rod where we've got a series of capsid proteins that bind to the genetic material and form a rod shape structure. Now, there's a variety of elaborations that can occur to these basic structures, but that's pretty much the appearance of the simplest viruses. Now, let's just take it a little bit further and note that in many viruses, that some of the capsid proteins take on some specialized roles, and we call them spikes, well, because they look spike-like, sticking up off of the surface. And we'll see that these are important as forming receptors binding to proteins on cells that the virus will be able to infect. As a matter of fact, the spikes on the surface of the coronavirus is what gave it its name when the first electron microscopic images of the coronavirus were inspected. It inspired the appearance. It caused the virologists to think that this looks like the corona of the sun. Not quite flaming, but certainly having that wisp-like appearance around it, and that's all of the spike proteins sticking off of the surface. We use a lot of 3D and artistic renditions of viruses to help us to better see what they look like, because, well, we can't see them in all of their true detail, even with an electron microscope. So we rely very heavily upon these computer-generated or otherwise drawn models of the virus. Now, one final elaboration, which we need to mention because it's going to be important, is some viruses, particularly animal viruses, do have a membrane around them. Now, this is not, it doesn't serve the same purpose as the membrane of a typical cell, and there's no cytoplasm inside. There's nothing really going on. There's a capsid inside, but some viruses just have evolved the ability to kind of steal membrane from the host cell in which they can put their spike proteins, the receptors. And this allows them to more efficiently infect potential host cells. And we're going to see that that's the case for the coronavirus. Take a sip of my coffee here. Okay, so let's look more specifically at the virus that causes COVID-19. And this is known as the SARS coronavirus type 2, and it's an enveloped virus. We're looking at it in cross-section, and there's a fair number of proteins present. We're not seeing them all, and some of them are only produced when the virus is replicating. It actually has sort of a rod-shaped capsid, even though it's very hard to perceive here because there is a protein that wraps around the RNA, genetic material, and that lies within a membrane, this phospholipid membrane, very similar, basically the same structure as a membrane of a cell because it's swiped from the cell during replication. But the virus now has inserted its own proteins in that membrane. So, and the spike protein is of particular importance because it is what allows the cell, allows the virus to infect specific target cells. And it actually binds to a couple different proteins, but one of them that's received a lot of attention is the angiotensin converting enzyme 2. Big name, ACE2. It's a membrane protein that is present on the surface of a number of different cells in the body, including cells of the lungs and respiratory tract. And the coronavirus has just evolved the ability to use this particular protein as its target for binding to and then being able to infect and attack and replicate within the cells that have it. All viruses just happen to use some particular receptor on their target cells that they've just evolved to bind to. So now let's take a look and see what happens when the virus actually infects the cell and replicates. So what we see here is sort of a series of time-lapse images, a series of steps of the replication process from the virus first binding to the receptors on the surface and then penetrating into the cell. It can do so through a couple different mechanisms. It seems commonly that the virus is actually endocytosed, we say. It is taken into the cell in a little membrane vesicle, which it then is able to rupture and then release its RNA genome. Or the virus, because it has this membrane envelope, can just fuse. It literally, the membrane of the virus becomes one with the membrane of the target cell, and in doing so allows its genetic material to be released into the cell. Now, this is another point where we can contemplate what it means to be a virus. Because at this point, the virus is nothing more than its genetic material. Everything else that was part of the virus outside of the cell is, for the most part, now no longer needed. Oh, there's a few exceptions, but for the most part, all of the proteins necessary to get the virus into the cell now are no longer needed, and the virus is just its genetic material, which is RNA in the case, and we're going to come back to talk about the RNA of the coronavirus. But the RNA carries the genetic information for producing all of the proteins needed to produce more viruses. And so once it's released into the cytoplasm, it takes over all of the cellular machinery to allow copies of the RNA and to make copies of all of the proteins needed to make new viruses. And then they all come together to form a new virus through a process called self-assembly. Now, this is another example of where being a virus is fundamentally different than being a cell. A cell replicates by dividing in half. A bacterial cell pinches in half, and then you have two. The cells of our body divide by making copies of their genetic information. Mitosis, some of you have heard of. And then the cell divides in half. Viruses don't divide in half. Inside the host cell, they produce all of the different components of the virus, which then spontaneously, by virtue of the properties of these proteins and the genetic material, spontaneously come together to form new viruses. I find it, one way of thinking about it would be to imagine that you had a car factory floating out in space, and all of the parts of the cars were floating inside this huge assembly building, and the car would spontaneously assemble. All those thousands of little parts would spontaneously assemble because they all would fit together in one particular way, and it would all just come together spontaneously. Well, that's the way viruses replicate. Now, I've got something I would like to show you to illustrate this. So I'm going to stop my screen share here for a minute. Okay, so here is a model of a virus. Now, this is in and of itself a multidisciplinary project. I need to thank my colleague, Dr. Craig Harold from the physics department for helping to make this model of a virus. And it is a self-assembling virus, at least I hope. And it comes apart into a series of pieces. I can disassemble this whole virus. I needed help from Dr. Harold to make this virus because even though it is a self-assembling virus, it required some assembly to actually make it. So I'm going to take this virus and I'm going to put it into this jar. Okay, now, while this process is a little bit noisier than what would normally happen for virus replication, it will illustrate how parts that can fit together in a single way will do so and self-assemble spontaneously. Makes a little bit of noise. Well, almost. One little problem with my container, because my self-assembling virus is held together with magnets, one of them stuck to the lid. Excuse me. Let's try that again. Sometimes getting the last one to fall into the right position is a little bit tricky. Self-assembled. So, likewise, all the various proteins and components of a virus will spontaneously come together by the fundamental properties that hold it together. Okay, let's go back. If there's any questions out there, I'll be happy to. Oh, by the way, the virus in the jar puts a whole nother meaning to the word of containment for a virus, I think. Okay, well, so let's press ahead and talk about some other things. So I want to take another look. As I said, we'll take another look at what viruses look like as they infect cells. Oops, let's get some control here. So what does a virus look like inside a cell? And for that matter, what does an infected cell look like? And so what we're going to do is we're going to look at a series of images at ever increasing magnification for cells of the lung, which is what the coronavirus infects. So we're going to look at a series of images. So this is a cross section of a typical lung, and we can see we've got various sorts of air ducts, bronchioli, and so on, and the blood vessels and so on. And then most of the bulk of the lung is made up of the small little alveoli that are the air sacs where oxygen and CO2 exchange occurs. So let's zoom in a little bit. So now we're looking at about a 10x magnification cross section, and we can see the cross section through the alveoli. Now, in a 3D model, all those little air sacs in your lung look like this, and of course you've got just innumerable numbers of these, and this is where the air enters and then the oxygen and CO2 exchange occurs. But we're just looking at those in cross sections here. And we've got all these cells lining, making up the borders of the alveoli. And so let's zoom in, slightly higher magnification, histological image stained in this case so that we can see the nuclei of the cells and the cytoplasm. But again, we're looking at just common light microscopy here, and so we'd never really be able to see the viruses at this magnification. So we're going to zoom in now using an electron microscope to see more detail, a single cell. And now let's take it up even further. And at about 10,000 fold, we can begin to see the coronavirus. And this cell, we're just looking at one small area of the cell, and you can see it's just full of viruses. It's become a virus factory. And it takes a much higher magnification to actually see the individual structure of the viruses with that corona type appearance. Okay. So where did COVID-19 come from? Well, where did the virus come from? And the best evidence we have is that it originated in bats in China. The coronaviruses are known to be common in a number of different wildlife, and bats is just one source. And through genetic analysis, we can show that the SARS coronavirus type 2 has about 96% homology with another coronavirus that had been previously studied from bats. As a matter of fact, it's got about 88% homology with the SARS coronavirus that caused an epidemic a few years ago. And so we know that these are all related. Now, how it got into humans, we do not know for certain, even though in Asia, in some cultures, they do consume bats. Nonetheless, it's possible it passed from bats to some other type of wildlife and then to an animal market, and then was possibly consumed by humans through that route. We don't know for certain how it happened, but we do know that the outbreak did occur in Wuhan, China, where these types of markets are known to occur. So why do new forms of the coronavirus emerge? Well, we need to take another look now at its genetic material. And as I said, cellular organisms, from bacteria to humans, we store our genetic material in DNA. DNA is our repository. It's the safe place for our genetic code. And I'm sure you all are familiar in principle with the double helix structure of the DNA molecule, which has two strands, one that carries the actual genetic code and one that is complementary, and it allows error correction to occur. It's one way in which if a problem, a mutation occurs on one base, it can always be corrected by matching it up with the proper complementary base. The double helix is one of the most remarkable structures for allowing faithful, almost completely error-free replication of information. But in order for the genetic code, the genes encoded by the bases of the DNA molecule to be utilized, it has to be first converted into an RNA molecule, which is very similar, except it's single-stranded, and it's got some slight difference in the bases, substitution of a uracil for thymine and so on. I'm sure many of you are familiar with the basic notion of RNA versus DNA. But the genetic code, our genetic code needs to be converted from a DNA form to an RNA form. And then the RNA sequence is used, it's translated, we say, into a series of amino acids that make up proteins. We've got about 23 or so thousand genes in our genome. Typical bacterial cell maybe has a few thousand. Coronaviruses have 26 genes, and they're held not on a DNA, but on an RNA molecule. It's a single strand. There's no complementary strand. It's a single strand of RNA, and that is then used directly to be translated to make the proteins that make up the coronavirus. Now, because it's RNA, that leads this virus to be much more prone to undergoing mutations. On one hand, when the replication of the RNA molecule occurs in the cell, since there is no complementary strand, if an alteration to a base occurs, there's no way to correct it. There's no backup strand that can be used to recognize that there's been an alteration to a base, to a base. So it's much more prone to errors. And when there is a change to the sequence of the RNA, that leads directly then potentially to changes in the amino acids that change the proteins, which then change the properties of the virus. And by changing the proteins, that can lead the virus now to infect other types of cells, or to spread more readily, to be more stable outside of the body. It changes the properties. Now, there's another way in which viruses are able to mutate and take on new properties. And that's by mixing their genes with the genes of a different form of a virus. And when a cell, and this typically might occur within a natural host, such as a bat, is infected by two different forms of the virus, their genetic codes can mix together. And we end up then now with a form of the virus in which genes from two different precursor forms have been combined together in new ways, which again can now cause it to be much more infectious to humans. And we do believe that this has occurred at some point in the evolution of the SARS coronavirus type 2, that recombination has occurred like this. So this ability of viruses, and it's their proclivity, I suppose, to mutate and get genetic information from other viruses, leads them to be much more prone to mutation and forming new forms on a regular basis.
Dr. Spilatro, we have a question that I think is related.
Okay.
So we have somebody who said they remember hearing in the news this controversy of whether or not the coronavirus, COVID-19, was natural or created in a lab. How did scientists tell the difference?
What they do is they look at the genetic code and see if the code compares to naturally occurring viruses. And indeed, when they look at certain parts of the genetic code of the SARS coronavirus type 2, what they see is that it's very similar to the code of other viruses that had undergone recombination in a natural, we know that was a natural occurrence. So, our current ability to do highly detailed genetic analysis allows us to track the evolution and the similarities between different viruses, so that we can with great confidence say where those viruses originated, and we can also, because we know that they do mutate over time, we can use changes in the code as a way to track when certain changes occurred. And thus we know that some of the key changes that have allowed the COVID-19 virus to become more infectious date back actually a couple years. This virus probably first began to emerge quite a while ago, and it's just taken it a longer period of time to enter human populations. So, the answer to that is the tracking of the history of the changes to the genetic code. Okay, great question. Fantastic question. Any others?
Nope, that's it for the moment.
Okay. Okay, well, I think many of you have heard about the symptoms of COVID-19, so I'm just going to quickly run through this and note that it causes principally respiratory tract infections, even though we are seeing that it can cause other symptoms as well, typically very mild symptoms in the vast majority of people. But in a smaller subset, we do have a greater incidence of more severe types of symptoms, including acute respiratory distress syndrome, which causes severe lack of oxygen and is what oftentimes requires people to go on a respirator. But also cardiac problems and kidney damage and other things that can be fatal in some people. I've got a video I want to show. I've taken a video from online and edited a little bit to get it to focus, make it a little bit more focused, and it shows something about the infection and how it causes some of those severe symptoms. So let's watch this.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. All the parts needed to create a new virus gather just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane. Now, With the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon. And when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells, and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs.
Well, that was a sudden change there. Okay, I don't quite know what happened, but at any rate, eventually the virus proliferates to the point where it can be fatal, as we had mentioned before. Okay, I'm looking at the clock and thinking about things that I want to cover and will have time to cover. And so I'm going to skip over a couple things, I think. And I'm going to skip over talking about the testing. And actually, I think this would be a good topic to come back to as in week 8. So I'm just going to skip over some things here. And this, and I want to say something about what our options are for dealing with the pandemic. And certainly there's been a lot of talk about pharmaceuticals that can be used to control the infection. Hydroxychloroquine, of course, have received all sorts of press, even though there's really no evidence that it is at all protective. A lot of interest recently in remdesivir. And, you know, these are some complex pharmaceutical chemicals that can actually help to slow the rate of replication and control the infection, but they do not, none have been discovered that actually prevent the spread of the disease. And what we need, of course, is to develop a vaccine to provide the immunity that people would have to prevent the infection from occurring in the first place.
So Dr. Spilatro, we have a question really quickly. It's actually
Okay,
a
great.
part question that is what you're talking about. And the student said, if the infection started in China, why didn't they try to contain it more so it wouldn't affect so many people in the US? And then the second part of the question is, it seems to be deadly to older people, but why is it so deadly, and why haven't we just become immune to it by now?
Okay, great questions. So let's begin by going back to China and why didn't they try to control it better? That's a tricky issue, and there's really two aspects of the way I would answer that. One of those is, to some extent, politics did get in the way initially of efforts to to recognize that this disease had emerged, and local health officials did not readily communicate with the World Health Organization about its emergence. So certainly there were some unfortunate political issues that got in the way. I guess we could boil it down to not wanting to be the bearer of bad news. But certainly once China did come to grips with the emergence of the disease, they did implement very rigid lockdown protocols and pretty much shut down Wuhan and a number of neighboring areas and cities, and in fact were able to significantly stem the spread of the virus. Now, whether that's going to continue to be effective, we don't know, and we don't know if it'll be effective here either. But after that initial hesitancy to deal with it, I think China did did take some pretty severe actions. Okay, what was the second question there, Suzanne?
So the second part was, it seems to affect older people and be okay for them, but why aren't we simply developing an immunity to it?
Well, and again, that's a two-part question. The first part, older individuals, mainly because older individuals oftentimes have so-called comorbidities, meaning other diseases, other conditions that leave us more susceptible to the infection, and likewise, older individuals have weakened immune systems. As a matter of fact, on certain accounts, one's immune system begins to go into decline somewhere around age 19 or 20. But certainly once you get up in the 50s, 60s and beyond, the functioning of your immune system is not nearly as strong, which leads you to be much more susceptible to a variety of diseases. Now, why aren't we just developing immunity? Well, we very well may be, and that's one of the big unanswered questions. We don't know enough about this disease to know whether or not exposure and development of the disease and then recovery leaves us with protective immunity. Do we become immune to it upon an exposure? Certainly that's true for certain types of pathogens, but we do not know in this case. And indeed, for those individuals who have only mild cases, it's quite conceivable that you recover from the pathogen, from the infection, but you do not retain, at least for a long period of time, the protective mechanisms, antibodies, B cells and T cells of the immune system that allow us to be immune for a longer period of time. Now that's part of it, but the other part is, at this point, only a small fraction of the population has actually been exposed. Probably at this point, we're looking at maybe 25, maybe at most. If we had better testing at this point, we might have a better answer to this question. But if people haven't been exposed to the virus, then they will not have the opportunity to develop immunity to it. And here we run into the double-edged sword. We want to prevent, we're doing social distancing to slow the spread of the virus, which helps to protect people, particularly people who are susceptible and likely to get the most severe symptoms. But that also means that the virus will not spread as readily through the entire population, leading to widespread immunity, so-called herd immunity. That hopefully will develop, but we know it depends upon the mutation rate of the virus as well. And I'm just about out of time here, but let me just note that the reason that we have seasonal influenza is because it's a combination of not everybody developing strong immunity and the virus mutating on an annual basis. And therefore, even if you were immune to the flu from last year, you may not be immune to the version that is circulating this year. We don't know what's going to happen with the coronavirus and whether it is going to mutate at a similar rate. And thus, even though people might be immune to the current version, we may not be immune to it in a future form. And so I will leave the discussion of vaccines to the future talk in 8 weeks, and that will be good because we should know more about the status of vaccine development at that time. So I'm going to stop my screen share, and I don't know if we are rigidly set at ending at 8:15, but I'm certainly open to any questions that anybody might have. Can we do that, Suzanne?
Sure, we can. We actually have one more question, and for those of you who do need to leave, we certainly understand the whole video is being recorded and we will put it up. For those of you who need to leave, I just remind you to please go into your Moodle classroom, take a look at the readings for today and for next week, and we'll be back next week, Monday at 7 o'clock, to look at media and its how it has dealt with the COVID and what it means to us to be critical consumers. So we have another question, Steve. The question is, if the exact cause had been identified early in the beginning, would it have been easier to find a cure?
Well, the sooner you know things, it's always better. But finding a cure, that's a tough issue. We do not have, the only cures we have for viral infections are one of two things. Either your immune system takes care of it, or you get vaccinated so you don't get infected in the first place. We have drugs that can treat certain types of viral infections. They lessen the symptoms, they slow the replication, but we really do not have any cures for viral infections. And certainly for some viral infections, once you're infected, that includes, the measles virus and so on. Once you're infected, you are infected for life, and it's your immune system that keeps the virus in check. So I don't know that identifying the cause sooner would have helped. I think it may have. What it definitely would have helped is putting in place the social distancing and containment mechanisms that could have slowed the spread and allowed slowed the death rate possibly. So, but I don't think at this point it would have led to actually any greater success finding a cure. Okay, anything else?
We have one more. If we knew about the coronavirus a long time ago, why didn't the National Health Department work on a cure or vaccine for it?
Aha. Oh, what a great question. And this is a great question. And actually, when I come back on week 8, I'll be joined with Dr. Greg Delamister from the economics department, who's going to be speaking in a few weeks about economic issues. And certainly an important part of the answer to that question has to do with economics. And we rely upon, to a large extent, pharmaceutical companies to develop vaccines. I mean, certainly there is a role for private laboratories in academic institutions and so on, but most of the real work in vaccine development occurs at pharmaceutical companies. Well, what's the financial incentive for a pharmaceutical company to develop a vaccine for an unknown or only a hypothetical pathogen, a hypothetical pandemic? This has been very difficult to stimulate. And so even where there's been opportunities for investing federal funds, taxpayer money, it's very difficult to commit money to a hypothetical. But maybe one of the lessons we're learning here is that we have to be more proactive because, again, once we're done, so to speak, with COVID-19, whatever that means, there will be another pandemic. Okay, they are inevitable, and maybe we might learn from this that we should be better prepared. But in our current economic structure, there is not enough economic incentive to make that type of investment. Fantastic question.
A shout out from Dr. Delamister, who says, thank you. He'll be sure to talk about the economics of developing vaccines. So.
Okay, fantastic.
And I think at that point, Steve, we are out of questions. Thank you so much for your time and insight tonight. It's a great place to start off our 8 weeks and gives us a base to begin on. And several people are popping up and saying thank you for everything tonight.
Okay, well, thank you. Great time. Enjoyed this very much. Okay, bye.
Bye-bye.